We said that these bases are going to pair up in such a way that we have a purine always bound to a pyrimidine. So your adenine is always going to be with thymine, guanine, and cytosine will all be bound together. Now the bonds between the bases are hydrogen bonds. And we said this before, excuse me, but hydrogen bonds are very weak, so they break easily, and that means they form back easily. So when these bonds break, in order for the DNA to be copied or to make a piece of RNA, once it's been copied, then those bonds can go right back and it goes back into its helical formation. So the pairing of A is equal to T, C is equal to G is the work of Erwin Chargaff, and it's called complementary base pairing. The feature that we're talking about allows the DNA to make a copy of itself readily, and also to be able to prepare to divide. Once your DNA is copied, then that cell is going to divide. If the cell's not meant to divide, it's not going to copy its DNA. So the whole point is each new cell is going to have a copy of the DNA of the parent. Now, in contrast, RNA is a single strand, so it doesn't have the double helix configuration. And it also is lacking or has a different base pair. It does not contain thymine. Instead, it contains a base called uracil. So when Erwin Chargaff was looking at RNA, everywhere he saw an A, there was a U. So the base pairing rules for RNA was A is equal to U and C is equal to G. Okay, once the structure of DNA and its relationship to the amino acid sequence was understood, they could start decoding the genes. And the first chemical techniques for DNA sequencing was in the 1970s, and they've refined it. So a lot of times, guys, what happens is when you have got something, it's wrong with you and you go to the doctor, a lot of times they can look, or certain diseases, they can look at the DNA sequencing and see if it was copied properly, and if not, that could be your answer. So there is a lot of diseases where they know that this base pairing has actually had what is called a substitution or a deletion, in which one of the base pairs was mispaired or deleted altogether from the DNA molecule, and this can cause a real problem when it comes time to make a copy of the DNA, because that in that point you're passing on faulty DNA, and that's not what is meant to happen. So DNA sequencing has really just come on since the 1970s. So they have worked out the human genome. It was called the Human Genome Project, and it was worked out. They know where all the genes are, many of them, and they have tons of data. They could actually take that human genome. It was worked out between, I think, like Harvard, Yale, Oxford in England. There were quite a few universities. It was a really, really large undertaking, and now they know that which genes are on which chromosomes. So they know, for instance, the breast cancer gene is on 13 and 14. It's called BRCA gene. And so now they know where they can go to certain chromosomes and look and see if that DNA has been damaged in any way or had any kind of mutation. Okay, bioinformatics, this is a huge, they use this a lot at St. Jude. It uses computer software to sequence the genome. So they can do a lot of assays and run a lot of panels on DNA using the bioinformatics method, using the computer software. Analyzing large sets of genes and comparing them to other genomes is called genomics. And then if you're looking at the protein level and the sequencing of the proteins, that's actually called proteomics. Sequencing of genes and their protein products is entirely, your genetics. It's on your background. I mean, whether you've got brown hair, blue eyes, I mean, it's all proteins, guys. It all has to be expressed. And you get that information from your parents. Linear sequences of DNA molecules are always going to be passed from parents to offspring. So later we will find out that if you've got a dominant trait and a recessive trait, that, on your DNA, that can determine which genes will be expressed. But we get 23 chromosomes from mom and 23 from dad, so, we're a blend. Molecular biology is measuring evolutionary biology, and it's very involved. My daughter actually has a degree in molecular biology, but you actually look at the DNA sequences of animals through time.